Alright, so we've already learned that for thousands of years people wondered about animal reproduction. And it was clear that the female egg was involved. After all, many animals begin their life by emerging from the female egg. But what was less clear was the role of males in the reproductive process. However, for thousands of years, people didn't systematically study reproduction. It wasn't until the 1700s with Spallanzani and others of his time that we see the power of science to uncover the mysteries of reproduction. So Spallanzani is going to use the frog as his animal model, in part because the frog undergoes a pretty straightforward reproductive process. So here we see the life cycle of the frog. The eggs are going to hatch into tadpoles. The tadpoles will go through metamorphosis, and that involves the loss of the tail and the growth of limbs, and then the young frog can crawl up onto land. Of course, it has to return to water, most species, for reproduction. In this picture, what we don't see is the male frog. So here's the female laying some eggs, and the question was, well, what is the role of the male frog? And again, for thousands of years, people knew that eggs were involved, but not what the males were doing. So just some terms here. Today, of course, we know that fertilization is the moment when a sperm cell enters the egg. But in the past, that wasn't clear. So in the 1700s, the time in which Spallanzani was working, the word fertilization is simply going to mean that the egg was somehow stimulated to begin development. So if these eggs hatch out into tadpoles, then we will say fertilization has happened. Now, under normal circumstances, fertilization was correlated with the presence of the male. And so here we see the male on top of the female. She has deposited some eggs, and the male is producing some semen. And because this happened outside of the frog bodies, this is called external fertilization. But again, the question is, what contribution does the semen make to fertilization? That's what Spallanzani is going to put to the test with some experiments. So if we think about a simple hypothesis that semen causes fertilization, well, we want to test that hypothesis with some kind of experiment. So here we have, if semen causes fertilization, and that'll be our experiment, then we should be able to make a prediction. So to test this hypothesis, notice it's sort of a form of A causing B, right? Semen interacting with the eggs is causing fertilization of the eggs. We learned in the past that one way to test a hypothesis that says A causes B is to block A to see if B happens. Spallanzani did that by putting little trousers, little pants on the male frogs to prevent the semen from getting to the eggs. So he was blocking A to see if B would still happen. Well, the prediction would be then, if semen causes fertilization and you block the semen from getting to the eggs, then you should get no fertilization. The eggs should not fertilize. They should not hatch out into tadpoles. And when Spallanzani did this in the late 1700s, he found that the eggs did not develop. So if the result is the same as predicted, then we can say the hypothesis is supported, that semen is a causal factor in fertilization. Now, at this point then, we have to address the issue of what is it about the semen that is causing fertilization. And you recall throughout history there were a couple of possibilities. Maybe semen is some material substance that is contributing to the egg in some kind of way. So it's the material aspect of semen, something physical in the semen that is stimulating the eggs to develop. Or maybe semen carries some kind of vital power, some spiritual essence, some productive force in nature that is stimulating the egg, right? And so we can kind of categorize this as the vitalist hypothesis. So to test these ideas here, whether semen carries something physical or some kind of power, spiritual essence or something, he did the following experiment. He had some semen in an open chamber, and then in a larger chamber he suspended some eggs. So these are unfertilized, frog eggs hanging above the semen, so there's no physical contact. And the idea was if the semen had some kind of spiritual power or vital power or something like that, it would have to rise above and interact with the eggs on the string here. So what he did was he allowed this arrangement to sit for a little while, and then took half the eggs, put them in water, half of the eggs put them in semen. And what he found was only the eggs that were put in fresh semen developed. If you take these eggs that were suspended over the semen without physical contact, they did not develop. So Spallanzani is going to conclude here that you need physical contact. The semen has to physically contact the eggs for fertilization to happen. Now it's possible if you were a believer in the vital force, you might defend your idea in the vital force. So Spallanzani's conclusion can be challenged. And one way to do that is to say, well, maybe the vital force can't move through the air. After all, this is air here. Maybe the vital force can only work if it's in fluid. After all, frogs are normally reproducing in the water. So maybe this particular vital force only operates if the semen and the egg are in water. Spallanzani wasn't persuaded by that type of challenge. He continued to believe that you need physical contact between the semen and the eggs for fertilization to happen. Okay, so at this point, Spallanzani says, yes, the semen has the power to fertilize the eggs, because if you prevent semen from getting to the eggs, you prevent fertilization. In addition, you need physical contact of the semen with the eggs. You can't just have any space between the semen and the eggs, so there didn't seem to be a vital force. But now we have to address the issue of the two parts that semen contains. After all, the semen is a fluid, but inside the fluid are these living organisms, what at the time were called spermatic worms. We'll just call them worms. And again, now that we have two components to semen, it's not clear which component might have the fertilizing power. You have to do an experiment. So think about what kind of experiment you would have to do to figure out whether it's the worms in the semen or it's just the fluid of the semen that has fertilizing power. If you think about that a moment, you'll probably arrive at the idea that somehow you have to separate the two, the worms and the fluid, so that you can treat eggs with either just the fluid or just the worms. Now, Spallanzani cleverly tried to do just that. Here we see one of his microscope slides with a drop of semen. And what he noticed under the microscope is that the little worms tended to sort of assemble on the outsides of the drop, leaving the inside of the drop, this space right here, free of the little worms. So this would be just the fluid. So he carefully, using a little pipette, sucked up a little of this fluid here from the center of the slide here and treated some eggs. And what did he find? They hatched out into tadpoles. They developed. So this fluid appeared to have fertilizing power. And from observations like this, experiments like this, Spallanzani concludes that the fluid is the part of the semen that has fertilizing power, not the little worms. Now, can we criticize Spallanzani, right? So we want to think about his conclusion, and we want to ask ourselves, well, is that the only possible conclusion? Might there be alternative explanations for his results? So think about that for a second. Can we criticize his conclusion here? And what you might have come up with is that, well, maybe it looked to him like this was just pure fluid here, no worms, but maybe when he was trying to get this fluid, he also picked up some of the worms, and that accounts for the fertilization of these eggs. So it seemed to him that the fluid was clear of the worms, but in fact it was not. So that would be an alternative explanation. It was a little sloppy here. Well, Spallanzani went on to conduct another experiment to get at this issue of which portion of the semen has power. Is it the fluid or the worms? In this famous experiment, what he did was he took semen and passed it through a filter paper into a flask, right? So we'll call that the filtrate. The stuff that comes through the filter paper will be the filtrate, and then he would use whatever comes through the filter paper, the filtrate, to treat some eggs. And he noticed that if you pass semen through one filter paper, the filtrate has significant power to fertilize the eggs. If you use 7 filter papers and pass the semen through, this filtrate has no power to fertilize the eggs. No development. And, of course, he didn't just use 1 and 7. He used 1 filter paper, then in another apparatus he used 2 filter papers, and then 3 filter papers, and 4 filter papers, 5 and 6 and 7. And what he noticed was that as you increase the number of filter papers, the power of the filtrate was reduced. The fertilizing power decreased as he increased the number of filter papers. Let's think about that mathematically. So here are some possible graphs that capture that result. So which of these graphs captures the result? So notice the x-axis has number of filter papers, so going from 0 to 7, and the y-axis is the percent of fertilization. So imagine you have 100 eggs, what percentage of them get fertilized? So higher percentage as you go up the y-axis. So here we have this graph, and if we practice our reading graphs, here we say as the number of filter papers increases, the percent fertilization does not change. It remains constant. For this middle graph, as the number of filter papers increases, the percent fertilization increases as well. For this graph, as the number of filter papers increases, the percent fertilization decreases. So of these 3 graphs, thinking about what happens as you add more and more filter papers, the percent fertilization went down. So that's going to be this graph here is going to be capturing the results of his experiment. As he added more filter papers, the percent fertilization went down. Now, how would you interpret these findings? This is a description of the result. Right? That's what happened, but why is that happening? Well, what we might say is that as you increase the filter papers, more of the worms are being trapped in the filter papers, and only the fluid is found down in the filtrate. And if that's what's happening, then you would conclude from this experiment that the fluid does not have power. Rather, it's the worms that have power. And you might test that by maybe just taking a sample of whatever's caught here in the filter papers and then treating some eggs. Interestingly, Spallanzani came to a different conclusion. Here's what he wrote about his results. If the 7 papers through which there has recently been filtered a sperm suspension is squeezed out into pure water where one puts unfertilized eggs, these hatch out very successfully, which proves that the filtration deprives a sperm suspension of its fertilizing power, insofar as the seminal liquid which it had contained remains upon the blotting filter papers. Then one causes it to come out by squeezing it. Believe it or not here, what he's saying here is that he still believes the fluid has the power. Here's what he did. Yes, he recognized that as you increase, here's one filter paper, as you increase the filter papers, this fluid, the filtrate, has less and less power, right? So that's what he was finding. But what he thought was happening was that the fluid was losing its power because of its passage through the filter paper. Now, how can he retrieve fluid from the filter paper? He would squeeze the filter paper out, so getting some of the drops of the fluid, and he would squeeze some of that fluid into some water, and then that water had fertilizing power. Now, he thought what he was squeezing out of the filter papers was just the fluid. So can you see another possible criticism of his conclusion? That what he thought was just fluid coming out of his squeezed filter papers may have actually also had some of the worms that were trapped in the filter papers. So Spallanzani Today, when we ask, how would you interpret what was happening when Spallanzani squeezed his filter papers, the fluid had fertilizing power, we would say, well, some of the worms also came out when he squeezed the filter paper. So in the end, Spallanzani was barking up the wrong tree here. Early in his investigations, he thought that on this one, he thought that the fluid had power. And so when he comes to do the filtering experiment, in his mind, he still thinks the fluid has the power. Even though, with our sort of fresh eyes and with historical hindsight, we can say, well, how silly maybe of Spallanzani, because after all, look, when you pass it through many filter papers, that's going to trap the worms, and the fluid alone will be down here, and that stuff doesn't have any power. Nevertheless, Spallanzani thought the fluid did have the power. The power was just robbed when it was somehow passing through the filter paper. So, he got it wrong on the role of the worms and the fluid. However, we can tip our hats to him for using the experimental method. Now here's another experiment that we'll consider here again to kind of complete the history of our knowledge of what it is about semen that is involved in reproduction. The spermatic worms that existed in semen were definitely alive, right? And so besides trying to physically separate spermatic worms from the fluid, another experimental approach involved killing the spermatic worms. This would be another way to block A to see if B happened, right? So here's our thought format here. If the living part of semen has fertilizing power and the living part of semen is killed by heat or poison or acid, then, right, the semen should lose its fertilizing power. And other people had done these types of investigations. When you kill the living part of semen, it loses its fertilizing power. The results were the same as predicted. So here's the interpretation that procedures that kill worms also deprive semen of its fertilizing power. And so one way to interpret that would be to say that the worms are the things that have the fertilizing power. When you kill them, you eliminate the fertilizing power of the semen. However, if you believe that the fluid had fertilizing power, how might you interpret those results? Well, maybe these procedures, like heat and poison or acid, yes, they might kill the living part of semen, but they also might influence the fluid, right, and destroy the power that was in the fluid. So there is a criticism of this interpretation, right? So again, one of the lessons here is that when you do experiments and you reach conclusions, you have to be mindful that there are alternate interpretations, and so you have to make arguments or do further experiments to either eliminate those possibilities and gain further support for your interpretation. All right. Finally, in the 1850s, George Newport sees a spermatic worm actually enter the egg, right? And we now call them sperm cells, and they are recognized as genuine sex cells. So in Spallanzani's time, in the 1700s, many people at the time did not believe that these little worms found in semen were involved in reproduction. Spallanzani actually thought they were parasites, blood parasites, had nothing to do with reproduction. Spallanzani thought fluid had fertilizing power. But in the 1800s, with better microscopes, people actually witnessed, observed these little worms penetrating the egg. And here is a sea urchin egg that has been fertilized by one of these little sperms, and when fertilization happens, a membrane raises up around the egg to prevent further entry of other sperm cells, and that is the moment of fertilization. So at this point in the study of reproduction, we arrive at some tentative conclusions here. The only physical link between generations of sexually reproducing species, like these frogs, for example, is the sex cells produced by each. Right? So what does this suggest about the location of hereditary factors? They are located in the sex cells. Now, here's a question. How could hereditary information be passed on to offspring without the parent losing its own hereditary information? That is, if a parent has 4 limbs and passes this trait on to her offspring, why doesn't the parent lose her 4 limbs? And here we might say, well, maybe these sex cells have a copy of this hereditary information. So whatever information was used to build the parent's body, maybe a copy of it is present in the sex cells. And that's why parents won't lose their information, the information to build their bodies when they make babies, because they're only donating copies of their hereditary information. So at this point, here we are. So hereditary information must be passed on from parent to offspring. Sex cells were the only physical link between generations in many organisms. Therefore, in these organisms, the physical basis of inheritance is the sex cell. So at this point, even though Spallanzani got it wrong about the fluid and the worms, we're at the point where researchers are focusing in on these sex cells, these physical cells produced by males and females. And when they unite, that fertilized egg has the power to develop into another offspring of that type.